This is the RTK kinase pathway. The first step of the RTK-MAPK pathway is the binding of a growth factor to RTK. Activated RTKs contain phosphorylated tyrosine residues, which are used as docking sites for adapter proteins. Transmembrane glycoproteins that are activated by the binding of their cognate ligands, and they transduce the extracellular signal to the cytoplasm by phosphorylating tyrosine residues on the receptors themselves by autophosphorylation and on downstream signaling proteins. RTKs, or receptor tyrosine kinases, are one of the large cell membrane receptors that also act as an enzyme. They play an important role in regulating cell growth, differentiation, and survival. RTKs form cross-linked dimers with each other as they begin to associate with each other. The activation is achieved through phosphorylation. As RTKs become activated, the gene transcription process becomes altered. When RTKs malfunction, cell growth and differentiation can be affected.
The next step is the binding of SHC. SHCs are a family of adaptive proteins that recruit signaling molecules. They take external signals and communicate them internally. SHCs are involved in many different cellular pathways, including growth factor, G protein, and hormone receptors. Deregulation of SHCs has been linked to diseases like cancer and Alzheimer's.
SHC binds to SOS, which promotes the translocation of SHC-SOS to the cytoplasmic surface of the plasma membrane, placing it in close proximity to RAS. In its inactive state, RAS is bound to GDP. However, when SOS is positioned close to RAS, a nucleotide binding site forms in the protein, and GTP is swapped for GDP, creating another binding site, this time for proteins.
MAPK is then recruited to RAS. After a series of phosphorylation and dephosphorylation events, MAPK activates. Then, through another series of phosphorylation events, MAPK activates MAPK and then MAPK. MAPKs, or mitogen-activated protein kinases, are involved in various cell responses and stimulus. A few of the many processes that they are included in are proliferation, mitosis, and apoptosis.
Phosphorylated MAPK can now move to the nucleus where it can be phosphorylated transcription factors. This acts as a genetic modifier by increasing the binding affinity for particular genes. MAPK pathways are involved in many important biological functions, including regulation of cell proliferation and survival. This has been an important pathway to study by cancer researchers because of this.